in details. In fact, this time is to show in details what happened here, a little bit in details and so on. And then I think that I will stop here, let you walking before going further. Otherwise, you can be lost. Okay? So do you have any question until here? Any question? I stop sharing maybe. Where? Okay.
So
There is someone. Okay.
anh lên trên này anh tắt mic xem.
Please switch off your microphone if you are not talking, so I can hear you now. Okay. What is the question?
I had two RF and I don't know if the RF4 is known or for positive, as you can see in the figure 1.
Yes.
Yes. So the RF4 it has 459 hits from plus.
Yes.
But it overlaps with the RF2, and I select the RF2 for downstream analysis.
Yes.
I don't know whether RF4 is known or for positive.
The question is if RF4 is known or false positive.
Yes.
Normally, I think that it should be known, but since we are not sure, in practice, some of the ORF can overlap. Okay?
Yes.
If they belong to the same operons, you see what I mean? The operons.
Operon, yes.
You see, so if they belong to the same operon, they can overlap sometimes between, you can have a lot between the end of your first gene and then the start of the next genes. Okay? So I should put here known, but since we are not sure, we can add maybe putative known. You see what I mean?
Yes.
So you can put putative known because we are not sure.
But how can I be sure? Is there any method or?
Yes, the only way is to proceed is to make a blast. If you make a blast with your open reading frame 4, and then did you find homologs with known function or not?
Yes, I found known function.
Okay.
100%.
With 100%?
Yes.
And then the size of those homologs, is it the same size as your protein?
Yes. Well, that part I haven't checked that yet.
So if the size of the ORF4 is similar to the size of your open reading, your homologs, you can be sure that it's a known one.
Okay. But if it's not the same.
I mean similar, plus or minus 10, something like that. Okay? It doesn't mean the same size, the identical size. No, similar. It can be plus or minus 10.
Oh, okay.
Okay, so if not, you can put putative known.
Yes. Okay, thank you.
And then in your writing, you should put all of those information. I mean, I check the size of the homologs, and then the size was not similar then I can conclude that it's known or repetitively known depending on what you get. Okay?
Okay.
You should always justify why you choose something like false positive or known or orphan and so on.
Yes.
It's all?
That's all.
Okay.
Thank you.
You're welcome. Working with this known. Okay. And then after that, you really need to fill that part to avoid red bond. Every time you find a red in your annotation, it means something wrong. Okay? So Pineapple should fill this part in order to get the translation of the open reading frame, and then in order to remove that red error. It's not good. So I stop sharing. Is it okay for Pineapple? I didn't.
Yeah, success. Okay.
Okay. So since I'm on this annotation, the Pineapple needs also to have the table legend, the title of the table. You need all of that. And then the figure legend, it's not on the top, it's on the bottom here. So I stop sharing. That is a group of proteins that share the common domain. And then from that, we can group them to a clan or to a homologous superfamily. Okay? So by the same time, when you proceed to your analysis with Interpro, if you are lucky, you can get repeats or you can get active sites of your proteins. Okay? So from active site, it's the most simple one. It's one amino acid. Repeats, it's a few number of amino acids. It means 5, 3 to 10 amino acid, and then you get a domain, and then the family, and then the superfamily, which it's in which you have a large family. Okay, and then if you have to choose, you should start with the domain and then move to the family and then move to superfamily. It means that domain is more precise than superfamily. So you can get a domain for, let's say, calcium ATPase, and then the family, it's the family of ATPase, and then the superfamily or clan, it will be with all of the ATPase, included the AAA ATPase, for example, for instance. Hope that is clear for everyone. Did you hear me? No, no. I have a message on chat. Salome said thank you. I suppose that she hear me.
Sorry, can you talk again? Some of students didn't catch the idea. So what is the question?
The question was, what is the difference between the family and homologous superfamily?
Okay, so class, please pay attention when teacher answer the question.
Yes. So it was a question. It was a question from Xiaomei on the chat. So in fact, in Interpro, you have several type of domain. I mean domain, or several type of entries. You have active site. It means one amino acid in your protein, if it's known. You can have repeats. It means few number of amino acids from 2 to 3 to 10 amino acids. If you find them in the protein that are repeated, some of them are really important for the function of the protein. So you have those kind of entry type in Interpro. You have the domains. It's a minimal. That means the domains could be 100 amino acids or 50 amino acids. So it's a minimal unit. So if you get several domains that they can belong to the same family, and then so you have family. For example, you can have family of calcium ATPase, family of proton ATPase, family of potassium ATPase, and then from the families you can group them to a clan, and then you have a clan of ATPase, and inside the clan of ATPase you have proton ATPase, calcium ATPase, or potassium ATPase, for instance. So from psi until organelle, it's from simple to the large family. So if you have to choose, you should choose, if you have domain, family, and superfamily, you should first choose domain because it's more precise. Okay? It's okay for everyone? Yes.
Yes.
I have go do beach. You say yes. So I should also share against I stop sharing this, and then I will share another screen. That one. Do you see my screen? No.
Not yes.
Not yet. Okay. I have it. Do you have it?
Yes.
Someone recognize its annotation? Someone recognize the annotation? No? No one recognize the annotation?
I
Yes.
think in the
Yes.
chat, yes.
Yes, it's written yes. Okay. But what I'm going to talk is, it's important for all of the students because you always make the same mistake. Here in the table 1, you have the size in amino acid of your proteins. Okay? And then I can see some of the size of amino acid are wrong. For example, ORF2 here. You see, this open reading frame is complete in 5', complete in 3'2. It means that, and then it starts at the position 2 and then end at the position 244. So if it's complete in 3', it means that it has a codon stop, right? And if it has the codon stop, and then you say that the size of amino acid is 80, it means that you count the codon stop as an amino acid. Is it clear? It's okay or not? So it means here that you should remove one amino acid because the codon stop is not the amino acid. So every time when your open reading frame is complete in 3' end, you should pay attention about the size of your amino acid. You do not count normally the codon stop as amino acid. But the size of your open reading frame in base pair, it's probably correct. 244 minus 3 plus 1 minus 2 plus 1. I'm not sure that it's correct. Very soon.
No, I think students don't count the nucleotide, the three last one.
No, they should. For the open reading frame in base pair, they have to count it because the gene is from the ATG until the codon stop. That's it, a gene. You need that. That's it, the features of the gene. But for the proteins, it's from the methionine, if they have a methionine, or the start, if they have a start, until the last amino acid, not until the codon stop. If you use the position of the codon stop to count your amino acid in size, it will be wrong. You will have one amino acid more. Codon stop is not an amino acid. Keep that in mind. That is a message. It's okay? So, I'm not also sure that this column is correct. The column about complete in 5 prime. In this case, I'm not sure that it's correct because here, if your open reading frame start at the position 2, it means that you are not sure that there is a codon stop before that position. Did you follow me? Yes? Everyone understand why I say that? It should be complete in 5 prime if you are sure that you have a codon stop before. The only way to be sure that you have the codon stop before, it's only if your start position is at least 4. If it's 4, it means the 3 codon just before it's a codon stop. If it's 5, it means the 3 codon just before the codon stop. If it's 6, it means the 3 codon before it's a codon stop. But if it's 2 or 1, I'm talking about the start position. If it's 2 or 1, you are not sure that you don't have the 3 codon before. So you are not sure that it's complete in 5 prime. So you should put no in that case. So in this line, you should put no in that case. That one is yes, yes, yes. That one is no, correct. And then yes, yes. And then you should check also about the size of amino acid. So hope that everyone get the point because it's important for your annotation. You should be able to find an open reading frame and then to find the features of your open reading frame. So I stop sharing this, my screen now.
Yes.
And then I put the die in the large screen. Okay. So now we are going to move forward and then talk about phylogeny. And then I hope that it will help you to proceed to the remaining part of the annotation and so on. So the first question is, what is a phylogeny? The phylogeny, it's only the relationships you can get between a group, okay, over the times. That group could be people, that group could be the eyes of the bird, that group could be. Here, it's a molecular sequences, okay? So, phyla, you will understand phyla, group or group. The group in Greek, it's phyla, or phylum, if it's in a simple way. And then we are looking for the true relationships between a group, and then this should be done over the time. And then to do that, you should have what is called the common ancestor, okay? So in the case of bacteria, the common ancestor, it's named LBCA, the general common ancestor. It's last bacterial common ancestor. We don't really know what is it, but it's some of the bacteria. It's the case of archaea, it will be the the last archaea common ancestor, and then in the case of eukaryote, the common ancestor is named Leca, Leca. Okay? So throughout the phylogeny, we can build an evolutionary trees, and then I will give you first the definition and why it's important to perform molecular phylogeny. So we need to perform molecular phylogeny because during the evolution, okay, biological process, as you can see here, define genes, define genome, define organism. Everything are moving, everything changing over the evolution. So it's important to study the relationships between them over the time, and then it can give you some explanation about why this kind of organism it's living in this kind of environment or not, why this kind of organism is it's able to be pathogen and so on, and in which kind of environment they belong to. And if you know where you come from, you can also maybe predict where you will be, how you will become in the futures or later. Okay, that is the reason why molecular phylogenics is important, to know a little bit or to study evolutionary relationship between genes and species. So when we perform molecular phylogenics, what we got, we only is the suggestion of the phylogenetic tree. That is called inferred tree. We don't really know what was the true tree. You understand what I see? The true tree. We only infer the trees, and then we hope that that tree inference really reflects what really happened during the evolution. Okay? So the tools you are going to use, keep in mind, only give you the hypothesis about the tree. And then this hypothesis is called inferred trees. Okay? And then we are going to see now some of the example of trees, phylogenetic trees. The first one is a tree of life. So to build a tree of life, you need a marker. The marker, it's a common genes, okay, or common non-coding genes like ribosomal RNA, or common protein genes between the set of species or the set of group of the proteins you want to analyze. In our case, it will be, they are common, they belong to a group because they are homologs. That's why we are going to build the phylogenetic tree from a group of homologs, okay? It also could be, if we are looking for the tree of life, it could be the ribosomal proteins, because the ribosomal proteins, you find it everywhere in each domain. Okay, you find it in bacteria, in archaea, in eukaryote, you find it in all of the species. You cannot survive as a species if you don't have a ribosomal sequence. Okay, the 16S, the 23S, the 18S in the case of eukaryote, the 25S in the case of eukaryote. Okay, to also build the tree of life, if you use proteins as a marker, it can be what is called informational sequence. Informational sequence, it's proteins it's used by the cell is biological process involved in information like recombination, like polymerase, I say recombination, polymerase, replication, and so on. Those proteins also are present in all of the species. So in that case, you can use it to build your tree of life. You can also use the core sequence. The core sequence are the sequence that are present in all of the spaces you are working on, okay, to make it simple. So if you want to explore the tree of life, you can go in this website, okay, and then you will explore looking in details what you get in bacteria, in archaea, and in eukaryote, and what are the relationships between all of the species. So now moving some of the example of eukaryote tree of life. So it's look like this, as you can see. You have nodes, we will come back on that. You have leaf here with the name of the species. They are grouped here by clade. They have metazoa, fungi, celiophora, and so on. Okay? We are going to come back about the number here at the nodes. They are called bootstrap, but we will see that later. So this is one of the example of eukaryote tree of life. And so now the evolutionary process, when I say to get a tree of life, it's a kind of resume of what happened during the evolution. What are the type of evolutionary process you can have for a genome or for a species? So from the ancestor, you can have, due by the speciation, genomes or species genomes. And during this process, from the beginning until now, you can have duplication of some genes, okay? It means that in the ancestor, you have one copy, and then on the species you get now, you have several copies of the same genes. It means that during the evolution, the process of expansion takes place. And then you get genes that are duplicated. You can also have the genesis of a genes. You do not have no genes in the ancestor, and we don't know why, but during the evolution, a new gene appears here that is called simply the genesis of your, You can have also exchange between the gene exchange between different species, okay? You have several species living in the same environment, and then during their living in that environment, they exchange. A genes move to one species to another one, and that process is called exchange. And then the symbol of that is AGT for horizontal gene transfer. You will also hear something like lateral gene transfer. Is the same thing. Okay, and then you can also have the deletion process of the genes. So it means between the ancestor and the species of your genome, the gene lost, okay, because it's become less useful, okay, or the gene have a lot of mutation, so that it becomes unfunctional or less functional, okay? So that is called deletion or loss of the genes. So what always you are going to build a tree, you have two types of trees, okay? You have what is called species tree. The species tree is like the tree of life. That is based on common marker, as I told you before. You also have the gene trees. The gene trees, it's based on the genes. It can be nucleotide sequences or it can be protein sequences. But many times, it may be useful to use protein sequences. Okay? because you get better alignment with protein sequences than with nucleotide sequences. We will see about the multiple alignment later. So keep in mind that you have species tree and then you have gene trees. And to interpret, to analyze gene trees, it's what you are going to perform. You need to have in mind how was the species tree. Okay? Even if you have only gene trees, to analyze it, you have to get in mind a little bit how the species evolve during the evolution. It's important to have it because you will know whether you have the duplication of the genes or whether you have the horizontal gene transfer of your genes. To decide whether it was a duplication or an horizontal gene transfer or a genesis of the genes, you need to have the species tree to know how the species evolve during the evolution. I move to you. So about the species tree, so I'm here at the species level. You can have one parental species, but several children species, or at least 2 children species. In this small tree with orangutan, gorilla, chimpanzee, and human, this is the common ancestor here. And then at this point, you have the time here, have something like the time. And then at this point, you have a speciation. Okay, it's called the speciation. It's when you have the separation between one species and another one. Okay, so here the species of orangutan appears, but the ones that will give gorilla, chimpanzee, and human continue. So the ancestor of gorilla, it's here where you have my mouse. And then at this time of the evolution, you have the speciation of gorilla. And then if you move forward further, and then you get here the speciation between the chimpanzee and human. Okay, that's why, due to the evolutionary tree, we know today that the chimpanzee is really close to the human. Okay, and then the orangutan or the gorilla is a little bit far. It's a little bit distant compared to the human. Okay, so and then inside the species tree, the gene tree evolve. Okay, is it clear for everyone? You have the species, the evolution of the species, but inside each species, you have the evolution of the genes. Okay, so each genes in each species evolve also. It's important. And then for that, you have 4 types of events. We already talked about some of them. The gene duplication, the gene loss, the speciation of the genes also, and then the lateral gene transfer or the horizontal gene transfer when a genes move to one species to another one. So it's only to show you how it's going. In yellow, you have the species tree. And then during the evolution, you see what happened here at this point of the time. Have a duplication happen. It means you have 2 copies of the same gene. And still in the ancestor of chicken, mouse, and human, it means that in these branches, as you can see here, I should have 2 copies. Everyone agree with me? In these branches, I should have 2 copies since the duplication happened here in the ancestor of chicken, mouse, and human. So if I move forward, you see I have a copy in red and then a copy in green, because I have two copies, but followed by a speciation. A speciation means that I have one copy in the ancestor, and then during the evolution, because I have this, I mean one copy of the genes, because I have one copy of the gene here in green, one copy of the gene here in red, due by the duplication here. And then during the evolution, there is a speciation between chicken and in the species tree and the ancestor of mouse and human. So you will have one copy of the green that will continue one copy of one gene copy, I mean, of in green that will continue in chicken, and another copy in human. That is a speciation. The same copy goes in one species and in another one. I hope it's clear for everyone. The same process happened with the red. I have one copy which goes in the chicken and another one in the ancestor of mouse and human. And then we follow what happened during the evolution. And then the red copy, if I follow the red copy, move. And as you can see here, in the chicken, during these branches, the red copy will lost in chicken. And then the red copy here lost in mouse. But the less copy continue in human. So what is observed today is only one copy in human. The question should be why we have only one copy in human? Simply because the function here is not useful in chicken or in mouse. Even if you have a duplication here, the way it's evolved during here the branch of chicken or during the branch of mouse, it's not useful for the mouse. So it decides to stop, to delete that gene, or to make it non-functional. It's okay? And then, but in human, it's still useful until now. So you found a copy of H1 in human. And then what about the green one? The green one has its own evolution models here, and then you find a copy in chicken, a copy in mouse, but during is here, the branch of human, you have another duplication, but only inside human to get two more copies in human. So at the end, you have the results of evolution. Okay, it's one copy of the genes in chicken, one copy in mouse, and then two copies here in three copies in human. As you can see, I have copy in green and then copy in red. Now, if I proceed to the phylogenetic tree, if I'm looking for homologs of those genes, I will have all of them, the green genes and the red gene here. It's okay? But if I get the tree, I will find that C, M, H3 and H2 group together, but H1 is alone. This tells me that probably C, M, H3 and H1 have the same function, and then H1 has a distinct function. Okay? But here, H1, H2, and H3 are three copies in human. Since they do not belong together, it tells me that something happened during the evolution, and what happened during the evolution is probably the duplication, since I have here several copies in human. Okay for everyone? So we are going to have some terminology. So an orthologs, it's only, or a gene family set, it's called homologous genes. If those homologous genes are called paralogs, if they come from a duplication like human 2 and human 3, okay, they are called orthologs. If they are coming from speciation, okay, it's clear for everyone. Paralogs, if they belong or they come from a duplication event here in red, you see the common ancestor between human 2 and human 3 genes here. Both are due by duplication event. Here, to call them orthologs, it's because they are due by speciation event here. If I'm looking for human 3 against chicken, the common ancestor human 3, you follow my mouse, here is a speciation. Since it's a speciation here, it means they are orthologs. Okay? So now my question is, human 1 and chicken 1 and chicken here, they are orthologs or paralogs? Human 1 and chicken, orthologs or paralogs? You write your answer in the chat. Orthologs, did you see the chat, Tom? I don't have it on my screen. Do you have some?
Yes.
You have some answer?
Not yet.
Not yet. Human 1 and chicken, orthologs and or paralogs? That's the question. Yes, no? Some of the answer? Yes, no? So if you follow the mouse, I have human here, human 1. I have chicken here. If I'm looking for the common ancestor between both, it's on this point and on this point, I have a duplication event. It means that human 1 and chicken, it's a paralogs. Someone say they are both. No, they cannot both. You are orthologs or you are paralogs. You cannot both, in fact. So, and then another one is chicken and mouse. As you can see, the common homologs here, if I'm looking for the common ancestor between both, the event that generate the two proteins from chicken and from mouse is a speciation event. It means they are orthologs. Okay? This is very important because, in fact, yes, it's very important because, in fact, orthologs has the same function Okay? But paralogs can have distinct function. If you have several copies of the same genes in a species, one of the copy will keep the ancestor function, and the other copy of the genes could diverge. The function could change over the evolution because you don't really need those functions since you already have one of the copy with the ancestor function. Okay? This is why paralogs can have different function, but orthologs has the same function. It's okay? So it means that if you are working on your own proteins and then you have several proteins in the same organism, and then you make a phylogenetic tree, even if they are duplicated genes, okay, you can know what is the function. Here, human 2 and human 3 are paralogs, but they do not probably have the same function as human 1. They probably have the same function as the one of chicken and the one of mouse, but not the one, so human 1 do not have the same function, in fact. It's okay? So that's why it's important to make a phylogenetic tree to confirm the function of your set of genes if you are in the case of duplicated genes, or to make the relationship or to make the function a relationship with other genes like here, chicken or mouse. So that was paralogs. We already answered the question. And then orthologs are more likely to have the same function. That is not the case of the paralogs. Okay. So what about lateral gene transfer or horizontal gene transfer? In fact, this evolutionary event causes a lot of incongruence between species and gene trees. Remember, I told you about species tree and then gene trees. So imagine that you have your species tree here with A, B, C, D, E, and F species, okay? That this is a relation tree between all of these species, so it's the species tree. So you take one of the gene 1, for instance, here, and gene 2. You found homologs, and then from the homologs, you built a tree, but a gene tree. Here, the gene tree is based on gene 1, or a gene tree based on gene 2. In the gene tree based on gene 1, as you can see, you don't have the same evolution or the same evolutionary trees as your species tree. You have A, B, F, C, D, E, or instead of A, B, C, D, E, F as in the species tree. What does it mean when you have B that is close to F? Because if the evolution is linear, I mean, if nothing changed inside the genes after the speciation of the species, if your evolution is really linear, I mean, everything happened in the same time in all of the species. You have mutation in the same way in all of the species. You have everything happen in the same way in all of the species. You will have the same gene tree as the same species tree, right? It's important what I'm talking about now. If your evolution is linear, I mean, everything happened in all of the species happen on genes, of course, in all of the spaces in the same way and in the same time, okay, you will have the same gene tree. The evolution of your species tree should be the same as your gene tree. But as you can see here, the tree of the gene tree is different of the tree of the species tree. And in particular, here in these branches, B have the same ancestor here as they have the common node as the F here. So if you find this in your tree, draw comparison with the species tree, it means that during the evolution, the B tree that was present in, the B gene, the gene 1 in B species, the gene 1 in B species here, move to the F species, and then continue, and then the evolution continue until now. And then if you have the B gene here, the B from gene 1, of course, the B here, which move on F species, you will find that the B gene here is very close to the F gene, since this one moved to that species. That's why in the trees here, they share the same common nodes. And when you find that, it's a sign of horizontal gene transfer or lateral gene transfer. In the same way, if you have gene 2 on C that move on A species, you will have gene 2 on C or gene A, gene 2 on A and gene 2 on C that share a common node here, a common one here. They are sister genes, and in that case, since it's completely different to the species tree, it's also the sign of a lateral gene transfer. It's okay? So, coming back here to get some key features about the phylogenetic trees, I should move this here. So, you can have two types of tree. The unrooted tree, or of course, the rooted trees here. So, in the tree, you have the leaf here, A, B, C, D. It's also called the external node. You have branches here, and then you have internal branches. Internal branches are those one. And then here, you have internal nodes, 2 branches, external nodes, which is your leaf, internal branches, internal node. So the difference between this is rooted tree. In the rooted tree here, it means you have a root. As you can see here, I have the same genes or the same species. Each of them has a root. We will come back on that. These are the two types of tree. As you can see here, it's better to have the rooted tree compared to the unrooted trees. So because in the rooted tree, the root is also called the outgroup, okay? In the rooted tree, you design, in fact, the common ancestor, and because you design the common ancestor, you know the signs or the direction of the evolution. Okay? You know that in the evolution, it starts here, and then we have the speciation between the group of CD and the group of AF, and then you move to get A, and then to get B, and then in other branches, you move to get C and D. Here, you don't know which is in the unrooted tree, you don't know what is the direction of the evolution. You don't know where and when it starts. Does it start here, here, or here? You don't have any information about that. And then you cannot get the direction of the evolution. You don't know if it goes like this or if it goes like that, and what really happened during the evolution. Okay? So in here, the summary is better to get the rooted tree or the outgroup of your tree if you can. So how many possible trees you can get from rooted or unrooted trees? Okay, for n sequence, the formula to get the number of sequences, that one. Don't worry, you don't have to learn that. Okay, so in red it's for the rooted tree, and then in blue is for unrooted tree. If you have only one sequence, of course, you have one rooted tree and then one unrooted. With two sequences, 3 possible rooted tree, only 1 possible unrooted tree. With 3, you see it's 15 tree, and then with 5, you see the number of trees you need to get, etc. So, as I told you at the beginning, the goal of the phylogenetic, of the molecular phylogeny, is to infer the tree and then hope that that tree is maybe the true tree. Okay? So, to infer the tree and then to find the ones that probably it's the true trees, you need to explore all of those possibilities. Okay? It's like for alignment, you need to explore in some case all of the alignment and find the one with the best score and the best value. Okay? So for the alignment, you have a tool called BLAST, who did the job for you, so that you cannot spend a century waiting for the results of your BLAST. Okay? It's the same for the tree. When you launch, you perform a trees, depending of the number of sequence you put in your analysis, you can have a lot of sequences. Since you can have a lot of sequences, you need a powerful tools to do that, to find the one, to estimate the one that can simulate the true trees. Okay? So, a phylogenetic tree, it's characterized by the topology, is the form of the tree, how many clades, how many groups, okay? And then the length of the tree, the length of the branches. We give you the rate of evolution or the time of evolution that happened from the common ancestor until now. Okay? So, to do that, it's a statistical problem, and you have tools to make you give you the truth with the topology and with the length of the branch. Only to give you that, to tell you that you have 3 main classes of phylogenetic methods. You have method based on sequences, the maximum parsimony and the maximum likelihood. And then you also have method indirectly based on sequences and probably based on distance. One of them, it's a neighbor journey. Okay, you will, during this annotation practical exercise, you will perform at least one tree using the maximum likelihood method and another tree using a distance method like neighbor journey method, okay, or bio neighbor journey method. So how those methods work, in particular here, I'm talking about the matrix method or the neighbor journey. It's really simple. The first thing, if you have a set of sequences, it start perform first the distance between each sequences. It's called the pairwise distance or the homology between each pair distance. And then from that, that leads to a matrix. And then you give the matrix to the method to estimate the distance between all of each sequence and give you the trees. Okay? I make it really simple. Okay. And then I told you that you can have billion of trees behind. Okay. And then the tools, in the case of matrix method, find the tree that have the much less distance. Okay, it's calculated the overall distance of, or the overall length of each tree, and then at the end, the tree it will give you, the inferred tree, is the ones which has the minimal length. If you sum up all of the length of all of the branch length. Okay? So the choice of the odd group, we will came later about that. It's better to explain you in practice how you choose it. But how I can say so that you can learn about it. How I can say that. Tam, how did you can you give me an idea about the central part of the Hanoi? How you call it? I remember the central district where our hotel when we come in Hanoi. How you call that part?
What do you mean? The central park in Hanoi, right?
Yes, the center quarter of the Hanoi. How you call it? With the lake.
The Hoàn Kiếm Lake.
Hom Kem Lake.
Hoàn Kiếm Lake.
Okay, Hom Kem Lake. And the ones we will call that part Hom Kem Lake. And the one of where the university is, it's how you call that part of the city?
Cầu Giấy.
The
Cầu Giấy is a district.
Yes, the district. How you call it?
Cầu Giấy.
Kobe. So I will say Ko. And then how come I will say Ko is better. Okay. And then you have, so you how to explain the choice. I'm looking for how to explain you more simple how we choose the on group. So you see, you have Vietnam. In Vietnam, you have the north, you have Hanoi, and then you have which city? Can you give me another city?
Maybe Ninh Bình. It's my province.
No, I will. It's better to check Hanoi. I will find Hanoi, so I will have it on the internet. Okay, Hanoi, where is, so I can have city of Hanoi and then everything. Not that wide. Map of Hanoi. It's because if I take the one of Marseille, it will not be useful for you. You will not understand what what I want to leave. Okay, I have it. Hanoi district, Hanoi district, and then I have Hanoi district, and then I have Vietnam, Vietnam map. I'm looking in the internet, in fact, to get everything for you. I'm looking for the nice map for you. I can send that one. Okay, I have a good one. Okay, but I will come back another time. But it's maybe because it's something that's sometimes really difficult for students. So I move this and then remember. So I come back on the lecture, and then I'm talking now about the out group and the in group. Okay, because if you choose the in group, you should also choose the out. Remember that the goal of that part of the annotaton is to get the origin of your fragment. Okay? And then for that, you make an hypothesis about what could be the origin, that is your in group, and then what could not be the origin, that is the out group. Okay? That is the root. It's clear for everyone? You follow me until now? Right. Okay. And then you perform your analysis, and then at the end, you should verify that your hypothesis is right or it's false. It's fine or it's wrong. In that case, you come back, you choose another one. So now I want to show you the role with a map of Hanoi and Vietnam, how you make that. Imagine that you want to prove that you live in Hanoi. Okay? I will stop sharing. Where is it? I stop sharing this. And then I move on. And then I share my website screen. Is that one? Everyone got it? Once again, I'm talking about in-group, but I take this image for you. Because after that, you will walk alone, probably a lot alone. And then it's better to understand the process. You want to prove that you live in Hanoi, okay? To prove that you live in Hanoi, you make a hypothesis that your in-group is in Hanoi because you want to live in Hanoi. In that case, Hanoi is a city. You will, by the same time, prove that you are not living in Hue or in Da Nang, because Da Nang is another city at the same level. It's okay for everyone? If you want to prove that you live in Ho Chi Minh, you need to prove that you don't live to another city in Vietnam, like Da Nang or Hue. It's okay? Now, if you want to prove that you live in Vietnam, this is your in-group. At the same time, you will not prove that you live in Cambodia or in Thailand because they are not a city but country. Okay, it's fine for everyone. Or I say it again. I'm talking about the choice of your in-group or your out-group. So, if, once again, if you want I'm at the level of city. If you want to prove that you live in Hanoi, Hanoi is a city, it's important to prove at the same time, to choose your out group, to prove at the same time that you don't live in Vinh or in Hue or in Ghana, because they are at the city level. If you want to prove that you live in Vietnam, at the same time, you have to prove that you don't live in Cambodia or in Thailand or in Laos, because they are country. If now you want to prove that you live in around How Come Lake, okay, in the level of district, I have to prove that, or you have to prove that you don't live in Koge, in Ko, okay? But I'm a level of district. So this example, in fact, show you that the level where I choose my in-group, it should be the same of my out-group. I cannot say, I want to prove that I live in Hanoi and choose my out-group as Thailand. That's impossible. It's two different levels. One, Thailand is a country, Hanoi is a city. It's clear for everyone. By taking that, you see them down, so you can tell me if everything is okay or not for them.
Do I understand?
They understand the philosophy? Okay, so, right. So I come back on my screen. It's that one. And then, so this is, so I have the screen. This is a PowerPoint now. So this is, I told you how to choose the out group. And then you have different evaluation methods. And we will see one of them. And most of them, of the evaluation methods, in maximum likelihood, it's called posterior probability, okay? And then you have also another one named bootstrap, okay? So how it's going, the bootstrap? Like in BLAST, you use, yes.
I think we should take a break, or
we should
a short break.
I have maybe only, I don't remember how many slides, how many, yes.
I think you have 15 slides left from this.
No. I have only 3 slides. No, don't worry about that, because the next, it's other thing. You see what I mean?
Okay, yes.
I have 3 slides and then you will have a break.
Yeah.
So I was here talking about the bootstrap. So when every tools in bioinformatics, you need something, or you need a score, a p-value, because it helps you to be sure that the results, it's consistent or not. In the case of phylogenetic tree, as I told you before, the way to be sure that your tree is consistent, you have 2 types of estimation. The first one, it's called the posterior probability. It's the probability or the confidence for each branches or each node. And then the other one, it's called the bootstrap, in which you test the topology of the tree. So I will give a small explanation about the bootstrap, how it's working. This is your initial data. Okay, you perform multiple alignment, and then from this multiple alignment, you get a tree. This is your tree on the right. Okay, and then what you will get, you will draw several, in fact, you will take several, pick several columns of your initial data and reconstruct, okay, several distance alignment. Okay, so you will have sample 1, sample 2, sample 3's alignment, you sample until sample 100 or 1000. From each alignment that have been built, that have been sampled from your initial alignment, you will construct another tree. Okay? So imagine that you have 100 more trees based on your sample, bootstrap sample here. If you found in this, on those 100 trees, 90% time, the same notes like this one, you will write here 90. Okay? It means that by chance, by sampling by chance your alignment, you get the same note, and in that case, it's make that note consistent. Okay? If over the 100 tree you get here, you get only one time that note, like that one, it means that this note here in your original tree is not consistent. So the role of the bootstrap is to tell you whether each node of the trees is statistically consistent or not. If it's less, if it's over 50, it means it's consistent. If it's less than 50, it means 50%, of course, it means it's not really consistent. Okay? And then this is an example of, this is not good. I don't know. I will call it later, someone. And then, so this is a bootstrap, a tree with a bootstrap value. So as I told you, if I have more than 50%, it's consistent, like here, 88. And if I have less than 50%, like 22 here, it means it's less consistent. It means I cannot believe to the group made by these branches, but I can believe here by the group made by this node in green. It's okay for everyone? And then a bootstrap value of 100%, it means it's a true node. Okay? It doesn't mean that it's a true node, but a good bootstrap value of 100% means it's a robust node. Okay, and then this part, we will do it in practice. We will come back on that. And then if you want to know more about the phylogenetic program and so, you have a link here with all of the phylogenetic tools that is used now in molecular phylogeny. Okay, so it's all for phylogeny. I hope that I tried to do not lost you. I hope that everything was okay. If you have question, I can answer it. Otherwise, you have.
Emmanuel, I am in this slide. So the slide that you introduced in the beginning of this afternoon. So normally, so we have the ORF and we choose the e-value beyond the threshold. We have the target, the max target sequence is 1000, right? So you perform the definition list and you have the table. And this table you have the several functions. So in this table is display the functional diversity. Okay, so the first before we perform the phylogeny, so it's nearly in the end. So we have to know which taxa that we have in the with the list of protein homologs, and we have to report like the how many of them that exist. So we have two tools. So the first, you have to perform the task report, and it will count for the own species and the family, the order and phylum. And then it will perform the summary of the reports, and it will have the task list. So, okay, if I move to this one, you guys been seeing in the screen? That's right. Okay, so now I am at rulebook. So over you have a tab with rulebook and a tab with the annotation. It's the way that you're doing with the annotation. It gives you quicker access to all the tools and like with information that you need to put inside. So when I go to this part, so we are finished at the class, right? So we move to the taxonomy report, the one that we have it here. So the first tool is the tax report. It already generates, it gives you the link on this one. And it's the internal tool inside the annotation website. Because if you see that plus, when you click on this one, you open to the NCBI plus. But if you click to this one, it's still inside. So it's internal tool for annotation. So they said that we have to put the plus hit, and it starts from sequence producing significant alignment. So if you already have the plus, so in the raw result, so you should make the copy from here. So we don't need to do plus again. And this is the result from the plus, again, the NR database. So you copy on from the beginning until the finished table. So before the alignment part. So for me, I have like 1,000 lines. When you make a copy, that paste it here. You have an example of the black heat here, and remember to include the environment sample. And actually, this one, for example, the unpunctual one, we are not going to study here. So that click here. And click on the taxonomic report. So this is the one that I got from the taxonomic report. Make sure that you copy all the raw data and put it in the raw result here. So you make the copy from beginning is here and make copy until the end. So I will normally it should be until here. Use so ignore the organism report. Can we continue? So when you are finished, so we have to create the table file. So where you have the different taxonomic classification. So you will start with the phylum, class, order, and what else do you have after the order? Class here. Family, and then the next one. Yes. So we have to make a list for this, and you see that we have in this raw result, we have, for example, this one is have 2 hits in the database. So it means that in the class, it found 2 types of the organism inside the class. And this one, we have 1. And you see the, sorry, I think this one. So they have the kingdom, and then the phylum, class, order, family, genus. You see this? So it comes from, and this is the species name. So now we have to summarize like this one. For example, all of them are from the Fimukush, and this city come from the in Fimukush. We have 1, 2, 3. We have 4 class, and inside this class we have 4 order. So they put like the e-value range from the minimum to the maximum. We have the number of results, so it means that the number of hits. And what else we have? So I think that's all. And keep in mind to that take the list of the taxonomic. Sorry, that take the list from the class that's above the cutoff. So you remember you have this one. You have the e-value threshold. So you have to take the one above the threshold or like below the threshold because the lower e value is better, right? So now we have to summarize this table and to give the table 5. So the tool is taxonomy list. You guys see this? So in this one, the example is start from the lookup, and you have to make a copy from that report, or you can, when you already put it in the taxonomy report raw data, you can make a copy from here. So in that case, that you don't need to rerun several times. So keep in mind that because it's an internal tool, so everything should be look exact like the example. For example, if we start from the lookup, it's meant to start in the lookup in the when you make a copy. So you cannot put like the bachelor at the status because it knows maybe the tool cannot recognize this one. And so if you want to make the table and report until the order, you take the number 4. If you keep at the family, you click on the family, the file. So this is what I got from the family. If you have a lot, you can keep the order. So this is raw table, and this is the table that we got from the. Okay, so another one. Không, em có tìm cái hít không? So you see in the table 5 is I think some. So this is table 5, and this is the table that we got from the task list. So the work is to turn this one to this. So for example, so here I have the virus and the bacteria. Very strange. So it means that I have to start from the kingdom. See 1 second with the virus. So normally I should check it again if it really the homologous with my sequence or not, because you see that for the more than nearly 1,000 hits, all of them are bacteria, but there are some strain with the virus. So, and for the case, if it's like if it only with the bacteria, you can remove the bacteria and then you start with the phylum. For example, if I remove the virus and I start with the phylum, so I have 2 phylum here. So the one is the Bacteroideae, and the other one is the Firmicutes. So this one and this one. And then you have to make like the class, the order, and the e-value range with the number 3,000. So turn this one to this table, and then you can remove this. So we will work until the table 5 and make sure you should have very nice table. And then we can continue with the in-group and out-group, because it's based on the table 5 to decide which is the in-group and out-group. Yeah.
Okay.
So this is until the family, right? But for some case, I get until the order. So there are a lot of people in the class, we have the same problem. And so why is give us only until the order? So normally from when we have some kind of problem, so we have to come back with the raw data. So this is the task report. And normally the task list, what I do, so it's if I start and I want to report with the, so if I take the family, the phylum number 2, so normally what it does is only cut, is been cut from here. So it will take the first one and the second one. So it cut on this line, and then it count for the number of the lines that appear for this one and this one. And in some cases that when you have non-report, so I guess it will be the unclassified. But we have to check, for example, if it, so we have this one. No, not it. It's not the way. Okay, maybe with the e-value is okay. So normally it's something like this. So I have the kingdom, the phylum, and then the the kingdom, the phylum, and then the class, and then it gives me the peptidase T, and this is the bacterium. So in this case, you see that they don't classify them to family. So it means that this one is unclassified. So when you have something like this, you can move to this one and you say that's the unclassified. So in this case, it unclassified at the family, and this is the unclassified at the order. And what about this one? So they are the bacteria, but they don't know if it's from which phylum. So this one is the unclassified as phylum level. And we have one virus here, and it's from this type virus. Only one hit, but the e-value is quite low. It's close to 0. And if I check for the alignment of this one, okay, right. So when you click on one, sorry, wait. So because I don't want to go back and do the plot again for the NR database and 1000 and check for this one. So the easiest to get, you just click on the sequence here, and then you check best select sequence in the plot format. And now I can do the plot with my ORF with the plus 14, and you see the align two or more sequence. So in this case, the query is my ORF. The subject is the virus sequence. So you will do only, you align 1 second to another one. So in this case, you don't do like 1 second again the database. So what can you tell from the alignment? Are they look like the same? So when we have the big character here, what does it mean? So it means that the query and the subject, they look like the same. And you see that the identity is 89%. The alignment score, they are very high. So what can we tell from this case? Yes. Yeah. But why the virus have the same protein as the bacteria, and only one virus has this one? So you see in the list that we so we have only one virus. and the other is on the bacteria. So this is a biology question. There no bioinformatics here. So we have one sequence from the virus. Because when you put something inside the result analysis, so you have to make the comment about this. So I heard some of you make the hypothesis that the gene from the bacteria is moved to the virus. Okay, sorry. So you said opposite. So it means that you said that the gene from the virus it moved to the bacteria, and that's why there are a lot of bacteria that have this thing. So which hypothesis is correct? So I think I will give this one for you to do at home and to make the hypothesis by yourself, because we will have very funny hypothesis on this.
A hit, in fact, it's an alignment. But for one protein, you can have several alignments. That's why it's, the number of hits is higher than the number of lines.